I'm going to talk today about cellular computing. And, okay. So, have any of you guys taken 270? East 270, yeah. So what I'm going to cover today covers the first 20 pages of this book in biology. All the rest of it we haven't gotten to yet. What's it?
Do you need dinner?
No. Sorry. So, I guess while this gets started. So, lately in biology there's been an advent of trying to control life and program living systems to do things we want them to do. And so you're not going to have an iPhone full of E. coli anytime soon powering your device. But the idea is that we can use living systems to address a number of both medical and industrial problems. So cells and bacteria are really simple input and output systems. So they're constantly sensing the environment, deciding if they need more nutrients or if they need to move or whatever, and then producing some sort of output. Bacteria are really easily controlled via genetic engineering, and again, there's a huge range of industrial and medical applications. So for instance, you can envision we could design a type of E. coli that you take in a pill, and it would scavenge your gut and look for cancer cells, and then some sort of readout would let us know that there was cancer there. Speaking industrially, there are biosensors that have been made that have a color response, and so if they're looking for a leak in a pipe or something, they can put these bacteria into the pipe, and with low concentrations of this molecule, the bacteria turn green, intermediate yellow, and high red. And so you can see how it, once we get these systems working, they can have a wide range of applications. And so to write a biological program, it's very similar to a computer program. You just need a couple of parts. So your code here is DNA, and you have the 4 base pairs, A, T, G, and C. So by putting those together in any combination, you can make a human or a tree. Then you also have the functions, so the input and the output. And what I'm going to talk about today are primarily the use of logic gates. And then finally, kind of the compiler, and that is the E. coli. So all of the nutrients and machinery that are going to help your program to run. And so today I'm just going to talk about a really simple gate, which is an AND gate. And so everyone here probably knows that these gates function by having both an A and a B input, then that will allow the output to be produced. And so I'm taking this, what I'm going to show you is data from an article that was actually just published this year in biotechnology, Nature Biotechnology. So the field is really young. And so there are some little parts, and these aren't really going to be too important, but all I want you to know is that the inputs, the ons are arrows like this called promoters. The stops are terminators, like T's like this. And importantly, the output is going to be the production of a green color. And so in this system, they take a couple of switches and they stick them together next to a green protein. And this is going to get a little technical, but all that you need to know is that once it's inside the cell, based upon an input, you get production of the A molecule and the B molecule, and then if it works, you get turning on of the green color. And so how that is measured is just by running the cell population through essentially a laser and looking for green. And so in this case, because it's an AND gate, we have input A and input B, we get A and B produced. That allows this switch to be turned on, and we get production of this green fluorescent protein, which just looks like this. So all your bacteria are green. And almost 100% of the cells have turned green. So not only is the switch really nice, really tight, so it can be turned on and off, but these cells retain memory. So these cells will stay green for up to 9 days after their switch is flipped. So that's pretty cool. So there's a lot of things that we can do here. And in this article, the authors then made all 16 logic gates. So there are NORs and ANDs and trues and falses, and then also kind of a little more higher level XORs or XNORs. And so the reason why I show you all of this is that we now have the capability of creating really intense circuits within a biological system. And so maybe you're thinking, well, okay, that's great, we have gates, but now can we create transistors? And in fact, as of an article published last week, yes, we can. And so I just think this is really incredible. And so I want to show a tiny little animation. This is from NPR. And so this is really, so in this case, So you have your protein moving along a switch here, right there, lighted up in teal. And the protein, protein goes and makes what they call the instructions, which is just RNA, and that ends up causing the switch to turn around, sort of a semi-transistor fashion. And then we can control the position of those switches using these scissor enzymes that are here. Is this making sense?
Vaguely.
Vaguely? Okay. Maybe I can slow down.
Well, I'm not entirely sure how it would manage to flip it around.
Yeah. Yeah, okay, yeah, so that's a great question. So these are enzymes, they're from viruses, and they're called integrases. And that's literally what they do, is they come in and they cause the DNA to flip over itself and cut out. And then it can be flipped back and put back in. And that's just cool biology. I had nothing to do with that. And so in the case of our AND logic gate here, they can have two sets of transistors, and then you need two different colors of scissors, essentially, to turn around and make it work. Does that make sense? So it's pretty cool, I think, and pretty neat. And so now that we have some of this capability, you know, what can we do with it? And sorry, I gotta go back to this. And truly, the possibilities are kind of endless. And so this field is moving really quickly. And another thing that I want to point out is that this field is led primarily by engineers, and it was started by electrical engineers. And so the fact that all you guys are sitting here and thinking about code and software and, you know, networks and all of that is really cool. And so I want you to take a time to consider how you can apply that into a biological system. And so the first entirely synthetic genome was constructed last year. You guys, some of you may know Craig Ventnor, who is a pioneer in the Human Genome Project. They made an entirely synthetic bacterial genome and then put it into an empty cell and then grew a synthetic cell, so that's quite incredible. There's also now been research done creating what they call a bio-Wi-Fi. So if you have a giant population of cells, can they communicate to each other and only specifically to each other? So you can imagine in that case, let's say you have a diarrheal disease, and you put in a bunch of E. coli that are meant to kill them, well, they're going to spread out over the gut. They need a way to communicate. And so I think that's pretty cool. There's also been initiatives to create essentially chemical wires. So bacteria of different kinds are next to each other. They secrete molecules that then turn on activity in the neighboring bacteria, which is pretty neat. There are also biological kill switches. So I talked about this at a different lightning talk. But basically, this is showing just a bunch of bacteria under a microscope. Each one of these little things is bacteria. And then when they turn on a kill switch, over time, you see there are less and less. So in here, these bacteria are engineered to have, upon an input, to kill themselves when they receive an input, which I think is pretty neat. And so not only can you have bacteria kill themselves, but there have also been studies where they have engineered people's immune cells, T cells, to go and kill cancer cells directly, which is really incredible. And so, oh yeah, and this is nuts. So I think it's crazy. So you know about how there are extinct animals, obviously, plenty of them. And so there have been efforts in the ecology spectra to engineer DNA that we have from, say, mammoths or dinosaurs or whatever, engineer them in a way synthetically so that we can bring them back to life. And so they are de-extinct, which is just really crazy. And so the only thing I want to point out is that the biggest downside is that this is pretty slow compared to computer electronics. So for this switch I'm just showing here, for it to turn on, they looked at it after 2 minutes. So that's pretty slow when you think about the gates that you guys are used to and the stuff in your iPhone and that sort of thing. But again, we're not trying to create computers out of E. coli, but to create medical applications. And so all that, I hope wasn't too fast, and I hope it made sense. But I really want to talk a little bit about kind of some cool stuff we could make. So are there any questions or thoughts?
What's required to what's the dev environment for trying to code these things?
Yeah, that's a great question. So ideally, working in a lab is probably the easiest thing to do. But the kind of machines that you need to make to just construct the DNA that you want, you could make in your garage for like $200. Or you guys could probably make less, so you get better at the hacking. And that's actually a good point. So they worry about people, terrorists, being able to quick make some crazy swine flu or something. It's not that easy, but it's simple.
What kind of research are we doing with synthetic biology?
That's a great question. Unfortunately, not much. So in my department is sort of the bacteria department, and we're in fact doing almost nothing like this. And then there is up here BME, or biomedical engineering, which is also not doing very much in Michigan. I personally would like to see that be more of an initiative. But we do a little bit of genetic engineering in our lab every day, making mutants or making, turning proteins up or down or whatever we want. So in some sense, we're doing the basics.
So are these isolated logic gates that you have, or can you chain them together? Can you do like clock signals, combinational and non-combinational? I don't remember what the actual combination was. But so can you chain them together?
Yeah, that's a great question. So that's what I'm not sure if I have
Was that the wires thing?
Yeah, exactly. That's kind of what the wire stuff is about. So you have one logic gate, and the output of that activates is an input for the next, and so on. And so, but more at a chemical level, so it's more broad. So theoretically, yes, you can. But again, some of these logic gate studies just came out a month ago.
Yeah.
So I would imagine they're in development somewhere in the US. Thank you, guys.